Okay, I'm ready to start talking about gluconeogenesis regulation. Now, we talked about the gluconeogenesis pathway. I outlined the pathway in a previous video. Now what I'd like to do is I'd like to talk about how it's regulated, okay? And that's important because all metabolic pathways need to be regulated. If these things were if these things were just allowed to do whatever they chose and run out of control, then there's a good possibility that you'd run into what's known as a futile cycle. So in gluconeogenesis regulation, we see a process of reciprocal regulation. Okay, and I have that listed right here. Reciprocal regulation. And what reciprocal regulation essentially means is that the molecules that regulate the gluconeogenesis pathway, either activated or inactivated, will be the same molecules that will be the reverse of the molecules rather that function for glycolysis. And that's exactly what I said here. The process of regulating the gluconeogenesis pathway involves reciprocal regulation, which occurs when the same molecule has opposite effects on a catabolic and anabolic pathway. So the catabolic pathway in this case is going to be glycolysis, because that's the breakdown of glucose. Okay, and the anabolic pathway is going to be gluconeogenesis. Anabolic, we're building up the molecule from smaller pieces. Okay, so with that said, that's what's going to happen. It's going to be the reverse, but they're going to be the same molecules, and that's why it's important. So, important reciprocal regulators for gluconeogenesis. Okay, so recall that there was something called fructose 2,6-bisphosphate. And fructose 2,6-bisphosphate was a molecule that upregulated glycolysis. Okay. It activates phosphofructokinase in the glycolysis pathway, even in very low concentrations. Well, keeping with the idea of reciprocal regulation, you can assume, or you can actually probably have deduced, that, fructose 2,6-bisphosphate is actually going to decrease or inactivate an enzyme in gluconeogenesis. And in that case, the enzyme is fructose 1,6-bisphosphatase. Okay, that's fructose 1,6-bisphosphatase. Now if we go back, I got my notes here, I can show you real quick that this is fructose 1,6-bisphosphatase here, and that's the conversion from fructose 1,6-bisphosphate to fructose 6-phosphate, so it catalyzes that reaction, and that's the loss of a phosphate, which makes fructose 6-phosphate. And it has, again, reciprocal activity. It upregulates glycolysis and, inactivates gluconeogenesis. Now, another molecule that has similar effects is AMP, okay? And that's another reciprocal regulator. It inactivates phosphofructokinase, hexokinase, it activates phosphofructokinase, hexokinase, and pyruvate kinase. Okay, so those were the three main regulatory steps in glycolysis. So it upregulates or activates all three of those steps because it makes sense. It's low energy conditions. AMP is the signal in the cell that says we're low on energy. Let's start going through glycolysis and making ATP. Okay? Now, you can imagine that this is going to have the exact reverse effect on gluconeogenesis. Because gluconeogenesis requires ATP, okay, and it requires 6 triphosphates in order to make glucose from 2 pyruvates, you have to have high energy levels. So low energy signal is going to tell you to deactivate or inactivate this pathway. And that's exactly what happens. So AMP inactivates the gluconeogenesis pathway. And in contrast, ATP will activate gluconeogenesis. Since high energy conditions are required for the production of glucose, ATP activates gluconeogenesis. But if we remember from the last couple of videos talking about glycolysis regulation, ATP actually inactivates glycolysis. It has negative effects on the pathway because there'd be no reason to keep making more energy, and that's what I was basically saying. So the next one is not a reciprocal regulator. It's actually regulated by whether or not it's produced, and that enzyme is the regulation of PEPCK here. Okay, PEPCK, and that's phosphoenolpyruvate carboxylase kinase, carboxykinase, something like that. PEPCK is what I call it for short, and it is whether or not, and basically this is based on whether or not it's produced in the first place, and it's produced in the liver and kidneys. So if it's made, then gluconeogenesis can occur and it's upregulated, or if it's not produced, then gluconeogenesis is not going to be able to occur. Now, to sort of sum this whole thing up, I basically just wanted to say that, overall it makes sense that the same molecules activate glycolysis, inactivate gluconeogenesis. And it works both ways. So what I mean by that is the molecules that inactivate glycolysis activate gluconeogenesis. Okay, that's reciprocal regulation. That's what we're talking about. And what does this do for the cell? Well, what it really does for the cell is it saves energy, and because you don't have to make more molecules. Okay, you can make this one set of molecules that works on both pathways, so there's no need to make a whole new set of molecules to regulate each pathway. Okay. And it's basically just highly efficient, and efficiency is important in living systems. So that basically sums up gluconeogenesis regulation.